So we have here the basic setup of the lungs. We have a larynx, we have a trachea, and we have our trachea diverges right around here. This is our carina, which is the floor of the dividing point, and into our two primary bronchi, into our right and left primary bronchus, a right and left main bronchus. And then it will diverge again into our secondary bronchi, and then from our secondary bronchi to our tertiary bronchi. And we're not going to move to quaternary bronchi. We're instead just going to go from there to bronchioles. Because it's not really worth it for us to keep calling them tertiary, quaternary, pentanary, sextanary. It's not really worth it. So all of these things are going to be continuing past this point with further and further branching. Now, at the ends of these branches, we are going to have eventually a terminal bronchiole and an alveolar duct, which leads to an alveolar sac with individual clusters of alveoli. Now, alveoli is the plural, and an alveolus is singular. And the rest of this stuff is effectively just clusters of alveolar sacs and air spaces. Now this space right here is that cardiac notch where you're going to fit about that fist-sized heart of yours. Now this is the lungs with all of the alveolar sacs removed. And this shows you just how much branching you have. All that fractal branching that we talked about in Unit 2 is coming back to haunt us. And here we have our pulmonary artery and our pulmonary veins. We're showing here all of this individual arteriolar and venule perfusion going on in the lungs as well. Now, let's take a look closer at our capillary-alveoli relationship here. We have our pulmonary arteriole branching and creating this capillary bed. And we have our venule, post-alveolar venule, coming off and recombining over here. So this is the network going on right here. This is a cut-through section of the alveolar sac. This is an interconnected cluster. They're not individual bulbs right here. They're not a cluster of individual balloons. They are actually, the walls themselves are interconnected, a honeycomb, as it were. Now, right over here we have a bunch of elastic fibers that interlace over each other and provide elastic recoil. Now this will become important later. About 50% of the recoil in the lungs is actually provided by the elastic fibers in the lamina propria around the lungs. The remaining 50% is provided by surface tension of the fluid inside the lungs. Now, this sort of behaves a lot like a bunch of rubber bands wrapped around a mylar balloon, if you will. And if you don't know what that means, this is the internet. Look it up. All right. Now, this here is a cross-section through a trachea. We have here a C-shaped ring of cartilage connected at either end by the trachealis muscle, which is a smooth muscle band with elastic fibers connecting either end. We also have thyroid tissue over here in the esophagus, and the esophagus over here, not in the esophagus, and the esophagus. Sorry if that wasn't properly enunciated. We have smooth muscle around here as well. This is the lumen of the trachea. We're going to close up now and show our pseudostratified columnar epithelium. Now, if that's a mouthful to you, that's fine. Just know that that means that pseudostratified, looks like that it has multiple layers, but it doesn't. It is just one layer. It is columnar in that it has, in that the shape of this layer of cells is columns. And it is ciliated in that there are cilia, like a shag carpet lining this, and it is epithelium in that it is the outermost layer of tissue in this organ. Now, it is also with goblet cells, the cells that are shaped like goblets. The purpose of this pseudostratified columnar epithelium with goblet cells is to provide protection, and also the ciliated portion is to create sort of a conveyor belt-like motion to waft up and out trapped particles of dust and debris that get trapped in this lung, and move it up and out of your lung tissue, up into your larynx, into your mouth, where you swallow it and it gets digested by your stomach acid. Now that's not gross, that's how it's supposed to be. What's gross is when you spit it out into the public and people have to see your icky spit. So that's your goblet cells here that are producing that thick mucus. Goblet cells produce thick mucus that, in combination with the seromucous glands over here, labeled as tracheal glands when they're in the trachea, and labeled as just submucosal or seromucous glands elsewhere, are producing a sticky mucus secretion that traps that particulate matter: dust, bacteria, invasive particles, scratchy sand substance, silica dust, those sorts of things. And that's what's creating that trapped stuff. Now, the goblet cells are there. In the trachea, you're going to have a greater concentration of goblet cells than lower down in your respiratory tract. In your tracheal gland, these tracheal glands, these seromucous glands, you're going to have a watery secretion. That watery secretion has the job of humidifying the air. You're going to be bringing the partial pressure of the air in your lungs to about 47 partial pressure to about 47 millimeters of mercury. No matter what the humidity in the air you are breathing in, that's about the partial pressure that you want in your lung air. So the work of the seromucous glands is to create that humid, watery secretion. Now, you're also going to have blood vessels in your lungs. Now, they have the same job as pretty much anywhere else in the body, to provide nutrients and remove wastes and toxins, but there is also some supply and demand for oxygen removal of carbon dioxide here, because these are working tissues, but then again, you have oxygen passing right around here, so the exchange rate is a little bit different for oxygen supply and demand here. Now, here we have a cut of hyaline cartilage, which is part of our C-shaped ring. Now we have the walls of the bronchus. I've got smooth muscle around here. Bronchus tissue is going to be constricting and relaxing here a lot. So this wrinkly shape of the tissue is resulting from a bronchoconstriction. If I had bronchodilation occurring, then this wrinkling would be unwrinkled. So this is not wrinkled in order to increase surface to volume ratio, as you would normally expect if we were discussing something in Unit 2 or Unit 3, but, or earlier in Unit 4 for plants. This is just a matter of, well, there's less space and I have more surface area to fit in that space, so it's got a wrinkle. That's just a byproduct of having more stuff and less space to fit it in. It's got a scrunch up. Now, this epithelium, you can't see what's in it, so let's take a closer look. Before we do that though, keep in mind, cartilage, right here, veins, and arteries, this thick-walled artery right there. We've got a whole bunch of glands and stuff around. Let's take a look again. So this is a close-up shot of our lumen over here, and our pseudo-stratified columnar epithelium with cilia and some goblet cells. We have fewer goblet cells in the bronchus than we do in the trachea. Remember, the lower down in your respiratory tract you go, the fewer goblet cells you are going to find. This is important because that thick mucus that you're secreting, you want to secrete less and less of that thick mucus the farther down you go, because thick mucus in your lower airways is going to get trapped and cause blockages. And if you block your lower airways and there is no air in your lower airways, you're not going to breathe. All right. Now then, this gland over here is another seromucous gland secreting watery secretions, still humidifying that air. We have connective tissues in more of this lamina propria, more of this smooth muscle. We didn't catch that cartilage in this image here, but we still have cartilage in the bronchus. Now we have a bronchiole. We are now past that tertiary bronchiole, right? We are now past that tertiary bronchus. We are now into bronchiole territory. We're not seeing cartilage anymore at this point. Now we're seeing a lot more smooth muscle. We're still seeing wrinkly epithelium. We're not seeing nearly as many goblet cells. We're seeing some arteries. We're seeing some veins. We are going to be seeing still that pseudo-stratified columnar epithelium with cilia still in here. And now we're into the alveoli. Oh, wait, we had smooth muscle. Yeah, I mentioned that already. Now we're in the alveoli. All I'm going to be seeing in this alveoli, I'm going to be seeing capillaries, and I'm going to be seeing airspace. And what I'm going to be seeing is I'm going to be seeing squamous epithelium in exchange for my columnar epithelium. Squamous epithelium are flattened cells. These cells are now flattened because I need a flattened surface for gas exchange. This is where all of that gas exchange is going to occur. I'm going to move air, oxygen and carbon dioxide across my respiratory surfaces, from the alveoli to the respiratory capillaries in my pulmonic circulation. And these capillaries are going to do that for me. All right? I'm not seeing cartilage anymore. Now, this image here is going to be showing me these alveolar macrophages, these dust cells over here. I'm going to call them dust cells in these in the alveolar space because they're no longer in the blood. They used to be monocytes. Then they migrated out of the blood into a tissue space, and now they are alveolar macrophages, also called dust cells. All right? I see a better image now of this flattened squamous epithelium. I'm not seeing any cilia here. So I'm seeing these dust cells instead eating up the debris that my cilia couldn't catch before it got here. So my cartilage in my trachea was in a C-shaped ring. Its job was to hold that lumen of the trachea open, allowing flexibility during inspiration and expiration. My cilia was there to remove trapped particles from the air, transporting them upwards, out of the pharynx keeping particles out of the air, out of the lungs, and getting those particles of bacteria out. My mucus glands and my goblet cells were working together, humidifying the air for those mucus glands, and the goblet cells secreting sticky, thick mucus to trap those particles. The smooth muscle was controlling the diameter of the bronchi at all levels, from trachea to bronchi to bronchioles. And I had elastic fibers throughout, allowing elastic recoil of the lungs. Now, this is a scanning electron micrograph image of an alveolar sac. This shows us that the alveoli are not smooth walled. There are holes that punctuate the alveoli and allow them to communicate with one another. These air spaces have a lot of holes that allow for communication and air movement from alveolus to alveolus. And here I have another scanning electron micrograph image of the cilia. And these cilia are going to be present in all trachea and bronchi and bronchiole levels. Now here I have a transmission electron microscope image of either trachea or bronchi, and I'm going to take a guess and say that this is trachea.
That was way too big.